Hello, everybody. My name is Margarete Odenthal, as we already told you. And in this seminar, I will give you an overview about our experience in NGS applications. So first, I will go on and focus on technical workflows, issues, and improvements, and then I will roughly tell you about our experience in data analysis, and then I will switching to some perspectives concerning the mitochondrial genome and liquid biopsies. Molecular pathology takes more and more center stage in clinical decisions, and due to the increasing knowledge of tumor driving or therapy-relevant mutations, we have an increasing number of NGS samples in our department. And in our department now we have more than 5,000 samples a year which are analyzed by NGS. But number and requirements are continuously increasing. So the close interaction with the clinician is therefore highly recommended, and in our clinic this is carried out by different networks. This slide shows you the need of information of certain mutations or other genetic alterations for the decision of certain therapies to treat, here in gastrointestinal tumors such as colorectal cancer, adeno or squamous cancer, or cancer of the upper gastrointestinal tract. And the same is true for lung cancer. New drugs are under development, and experience of some drugs might be transferred to lung cancer. So all this makes it necessary to test a panel of genes to estimate drug resistance or sensitivity to treat the patient individually. So I think that targeted sequences is here the right tool to answer all the requirements of the clinicians. So we focus on genes of or loci which are needed to make a clinical study or to answer the clinical related decisions. And targeted sequencing benefits that only clinical relevant data are yielded, and it is much more sensitive, it's less cost effective, and it's also less sample consuming. So there are two approaches of targeted sequencing. One is the hybridization-based capture approach, and the other one is the multiplex PCR approach. Here in the hybridization capture approach, the targets are captured by hybridization of target-specific probes. And in this approach, by multiplex PCR, we enrich the targets by PCR. So in general, hybridization-based capture methods need more DNA than the multiplex PCR approaches. So when you have quite plenty of DNA, for example from fresh frozen material, or also sometimes from big samples of formalin-fixed tissues, then the hybridization-based capture approach is working. But sometimes you have only very, very few DNA, for example, when you want to perform single-cell analysis, or when you want to analyze cell-free DNA, then you have only low amounts, and then you can introduced one additional step of a whole genome amplification, and so you have amplified your DNA amounts, and then you can use it for hybridization-based capture approaches or also for multiplex PCRs. In our department, we are using cell-free DNA and also formalin-fixed or DNA from formalin-fixed tissues, and therefore we are dealing with the multiplex PCR DNA enrichment. So most of the tissue and cell material what we are processing in the pathology is formalin fixed, and that having the advantage to be the formalin is worldwide used and to provide an extremely high quality of histology. But it causes covalent links of proteins and nucleic acids, and that leads then to high fragmentation of DNA or RNA, and that is mainly due to the mechanical forces during isolation. And here you can see for RNA how the size of RNA is decreasing during the fixation time. Here you have only very low molecular weight molecules. In addition, formalin causes deamination of cytosine resulting in uracil, that is shown here. And then during the PCR, the C is changing in T because the C is recognized as from a U, and then you have C to T changes or D to A changes. And that is these transitions are also the most common artifacts when PCR-based analysis is performed on formalin-fixed material. And these artifacts can be removed from sequencing results by treating the DNA with uracil glycosylase before application to PCR-based analysis. So, due to the fragmentation and the different quality of DNA from formalin-fixed material, we highly recommend to quantify input DNA not by absorbance measurements, in which you measure everything which absorbs at 260 nanometer, and also not by fluorescence measurements by which the quality of DNA is not considered, but by quantitative PCR in which you determine the PCR accessible DNA, and that might be in particular important when you establish a new target panel or for a certain tumor set. So using a standard curve, we apply then 10 nanogram of the target enrichment in the target enrichment step. And this 10 nanogram is equal to 3,000 genome copies, representing then 1,500 cells, and that is quite a good number to represent also the tumor. So using the QIAGEN read workflow, 4 primer pools are applied, generating overlapping amplicons, and this ensures nearly 100% or very often 100% coverage of the target regions. And then the whole workflow is here shown, and you see that there are quite a lot of purification steps were performed, and that is done to get rid of primers, nucleotides, adapters, and so on. And so we perform a purification step after target enrichment PCR, after adapter ligation, and also after the final amplification. And in our lab, we perform this by automate, and that might be established by different platforms, also by the gene reader system of QIAGEN, which is also very useful. And we observe that automatization is less error prone and results also in less contamination between the samples or also between the runs. So in particular, when we establish a new panel, we always do quality control steps. And here the quality control is shown after adapter ligation, and you see here the samples after the first purification steps, after the enrichment, and then after the adapter ligation, and you see here very nicely the shift of the size when the adapter are ligated. And we are performing this by with the QIAXL capillary electrophoretic system, which is an automatic loading system, which is quite convenient. So here we had the opportunity from QIAGEN to test a new workflow, and in this workflow, the adapter ligation step is carried out only by one step. And we think that this method is extremely attractive because it saves time, that is one thing, and also it saves the procedure of handling the amplicons. And we are always we always try to avoid multiplex amplicon handling because we have a very high throughput in our labs, and therefore we are always afraid to get putative contamination by the amplicons. And so therefore we think when saving handling of amplicons, that is a really attractive step. So we applied this one-step protocol to two panels, the clinical relevant panel of QIAGEN. This is a panel covering 24 tumor-relevant genes with high mutation rates, and it includes 602 amplicons. And we used also a customized gastrointestinal tumor panel. It was designed by a colleague of mine, and it covers 8 gastrointestinal relevant genes and also some short hairpin repeats. So using these both panels, the read depth of the one-step protocol was slightly lower compared to the gene read core protocol, so the conventional protocol. However, the read depth was still quite high, so we had a read depth, the mean read depth was of around 2,000 reads per amplicon, and that is really quite nice. Then we compared the mean coverage of each amplicon of the one-step kit and with the core kit, and you see that we have really a very high correlation and also a significant correlation of the read depth. In addition, we looked for the clinical relevant mutations, and they were all identified by the one-step workflow, and they were also detected in the in the same frequency as in the core with the core kit. So we had here also a very high correlation. So in the past, we worked with different cancer-related panels of QIAGEN, I mentioned already the clinical relevant tumor panel, but also we used the tumor actionable panel, focusing on therapy relevant gene loci, and we used the BRCA1, BRCA2 panel, which I will introduce to you at a later point in my talk. And we used a prostate cancer and a liver cancer panel. And in prostate cancer, there is not much known about therapy-related mutations, and the therapy-related analysis is not yet well established. So we analyzed now 159 prostate primary tumors of prostate cancers, and we found that the most frequent variant was a variant in the KMT2D gene. This is also called MLL2. It is a lysine demethylase of histones, so functioning as an epigenetic writer. And then we also found a lot of mutations in TP53 and in the APC gene. And interestingly, our study showed that P53 and APC mutations are often associated with each other, whereas GLI1 and the SPOP mutations are mostly exclusive. And additionally, I would like to mention that activating mutation in the PIK3CA gene and in the androgen receptor gene was found, and they are highly therapy relevant. So the androgen receptor is a nuclear receptor functioning as a hormone-dependent transcription factor, and gene alterations such as gene amplification, point mutations, or splice variants are restricted to metastatic and castration-resistant prostate carcinoma. And the mutations in the androgen receptor lead to androgen receptor activation, which in turn results in resistance to the anti-androgen hormone therapy. And in our study, we found typical androgen receptor mutations which activate this transcription factor, and we always validated our mutation when we use a panel for the first time by Sanger sequencing. So another interesting mutation is the mutation in this POP gene, and this protein participates in DNA double-strand break repair, and mutations are associated with genomic instability. And this POP plays a critical role as a tumor suppressor and promoting the turnover of the SRC protein by interaction with this MATH domain. And all mutations we found were in this domain, destroying the interaction with this tumor suppressor, which in turn regulates again the androgen receptor transcriptional activity. So these mutations are quietly therapy relevant. So as I mentioned, when we establish a new panel, we always perform Sanger validation, and that is done because filtering of false positive variants is sometimes really quite tricky. In my lab, we are working with the Biomedical Genomics Workbench as a bioinformatic tool. And here, this workbench performs read mapping, annotation, primer and adapter trimming, and also the identification of SNPs in in different databases. So from a table of variants, you can interactively switch to a gene read view and showing then the region of base exchange and also the region or the amino acid track where you can see then the amino acid change. And that is quite nice, I think. By the Biomedical Genomic Workbench in a first step, also all technical filters taking place. So the thresholds of the forward-reverse read balance is set, the read count threshold is set, the frequency which is considered is set, etc. But in my experience, I think it is also very important to filter hotspot artifacts. I call them like this, and these are artifacts which are chemistry or system-specific errors. They are occurring then nearly in all of the samples with the same low frequency. And so you have also to define this. And then you have to do the functional interpretation if the variant is pathogenic or not. And all these steps lead then that you come maybe, for example, from 100 variants to one certain and also relevant variant in your sample. And QIAGEN is now offering here a whole pipeline from mapping to filtering to data interpretation. And this is done by different tools. First, as I explained to you already, the Biomedical Genomic Workbench, and then the functional analysis is performed by Ingenuity Variant Analysis, and finally you have a quite nice performance for data interpretation. And at the moment we are testing all these tools in our laboratory. So the clinical insight tool might be linked also to your in-house sample tracking system, providing then the sample-specific data such as cancer type, sex of the patient, etc. But most importantly, it says you which mutation occurs and also interprets if this mutation is pathogenic or not, and also tells you how the frequency is of the mutation. So this might be not so important when an NRAS mutation is occurring, because most of the people know that this mutation is pathogenic. But if you have unknown mutations, it's very helpful if you have this link to these big databases. And in addition, there is the opportunity to get the interactive links to publications dealing with the mutants of interest, and also to get information about present treatments and options. So up to now, my experience is mainly only with the Biomedical Genomic Workbench, but as I just told you, the other tools are just tested in our laboratory. And dealing with the Biomedical Genomic Workbench, I just would like to introduce you to to the simultaneous detection of mutations and deletions of the BRCA1 and 2 gene. So the BRCA1 and BRCA2 gene proteins are involved in DNA repair and are therapy relevant for treatment of ovarian cancer with PARP inhibitors. However, the genes cannot only be inactivated by mutations, but also by large deletions covering sometimes more than one exon. And up to now, these deletions are identified by multiplex ligation-dependent probe amplification assays. These are assays based on a probe linked to PCR amplification. In order now to identify mutants and deletions, we use the Biomedical Genomic Workbench tool. And for this study, we used 51 formalin-fixed biopsies with known germline alterations of the BRCA1/2 genes as a reference cohort. And 11 were shown by MLPA assays that they have also deletions. And then we used the next generation sequencing approach linked to the biomedical genomics workbench tools to look for copy number variation of the BRCA1 and 2 regions. And here you see one example. Here in the x-axis you see the different amplicons of the BRCA2 gene or the BRCA1 gene, and also the signal height of the sample. And here also the confidence interval is also shown. Yeah, but in black it's a signal of the sample. And you can nicely see that you have here BRCA1 exon 8 deletion. And that was also confirmed by MLPA, and we had in this reference material 100% concordance of these 11 samples. Then we screened further a bigger cohort of nearly 200 samples, but these were sporadic cancers of ovarian. And so we screened by NGS, and then we confirmed selected samples by MLPA. And here, not in all samples the MLPA approach was working, and also not in all samples the NGS copy number variation analysis was working. But the bioinformatics tool was working very, very nice. The problem was here that we had sometimes an imbalanced amplification due to DNA quality differences. And this imbalanced amplification we had in both methods. However, the copy number variation analysis itself were perfect and showed clearly here in this sample, for example, that you didn't have any deletion, but here the whole BRCA2 is deleted, and in this sample we have a deletion of BRCA2 exon 8 and BRCA1 exon 7, and in this sample half of the BRCA2 is deleted. So this worked really very nice. I want now to switch to another focus of our ongoing work, and that is tumor tracking and evolution analysis by ultra-deep sequencing of the entire mitochondrial genome. The mitochondrial genome is a small circular DNA molecule of around 16 kb, encoding polypeptides of the respiratory chain complex and structural proteins. The mitochondrial genome is highly susceptible to oxidative stress, and is then damaged and has an up to 10-fold higher mutation rate than the nuclear DNA. And that is due to the high rate of reactive oxygen species in the mitochondria, the less protective histone-like protein levels, and the limited DNA repair. So, after sporadic mitochondrial damage and mutations, the mutated mitochondrial genome will expand and replace the wild-type genome. And due to a selective advantage in, for example, replication and maybe also other mechanisms which are not totally understood, the mitochondrial genome undergoes an intramitochondrial expansion and also intercellular expansion, so that nearly after a while all cells carry mitochondrion with a genome which is then mutated, and that we call homoplasmic mutations. So due to the homoplasmy and the high mutation rate, the mitochondrial genome is an ideal target for tumor evolution analysis. And therefore, we established here workflow. So first, we established a multiplex PCR approach, and in agreement with the overlapping QIAGEN primer design, we designed also our primers and included then 108 primers and analyzed with these primers, which also generating overlapping amplicons, the whole mitochondrial genome. Here you see a homoplasmic mutation, which was then also validated by Sanger validation. In order now to test the tumor tracking by the mitochondrial genome, we used DNA samples from different lesions of non-small cell lung cancer, and these lesions showed a different tumor growth by histology. And we used a second cohort also with heterogeneous lesions, and this cohort was previously described by my colleague Stefan Schäfer, and he analyzed already all nuclear variants in this cohort. And when we now look how the mitochondrial variants are occurring, you see first here in red labeled that some mutants are occurring on all lesions of one case, and others are only private lesions here in blue labeled. So some of the tumors can be recognized to be monoclonal, having most variants in common, and other tumors are polyclonal, having none variants in common, having different variants. So in conclusion, whole mitochondrial genome analysis gives us the opportunity to follow up some tumor cells, their clonal expansion, and also we can determine if lesions are monoclonal or from different sources. So we used also this comprehensive mitochondrial genome analysis on a cohort of hepatocellular carcinoma, and we compared here the mutants also with the peri-tumor lesions. And here we observed that the mutations are accumulating after tumor development. As you see here, that much more mutations occurring when the hepatocellular carcinoma have developed. And importantly, we could show that mitochondrial alterations precede the nuclear genome alterations, as you can see here. This sample is these samples are all from one patient. This is a non-tumor sample, and you have already here in the respiratory in genes of the respiratory tract, you have already mutations, but no mutations of the nuclear genome. So our hypothesis is that expansion and accumulation of the mitochondrial mutation in the respiratory chain might cause further oxidative stress, increase the ROS generation, which in turn might trigger then the nuclear genome alteration and promote tumor machinery. So therefore, we think that the mitochondrial genome might also be used as a marker for inflammatory or oncogenetic features, and disease-related DNA, in particular tumor released DNA, is released from the tumor cells after necrosis, apoptosis, or maybe also by secretion. And therefore, tumor cell DNA is suggested as a novel target of therapy, follow-up, or diagnosis of tumor progression. Due to the high copy number of the mitochondrial genome and the high number of mitochondria in the cell, the homoplasmic occurrence of the mutants in the mitochondrial genome might be ideal as a circulating marker of tumor growth. However, this is a nice perspective. Until now, we are dealing only with the identification of nuclear genome alteration in cell-free DNA. But the challenges are here: low yield of the DNA, fragmentation and degradation of the DNA, and in particular the background of genomic DNA derived from non-tumor cells. And therefore, we recommend to use cell-stabilizing blood collecting tubes, such as PAX genes from QIAGEN. And these tubes have some ingredients to stabilize the cells in the blood, and therefore you can use much more time before you prepare the plasma samples. DNA extraction can be automatically performed on the QIAsymphony platform, and here you can use a 4 milliliter plasma format or a 2 milliliter plasma format. That is depending on your sample size. In our lab, mainly the EGFR mutations in small cell lung cancer are presently studied. In the EGFR gene, mutations of the tyrosine kinase domain are highly affected by mutations, and these mutations affect also the therapy with an anti-EGFR antibody or the tyrosine kinase inhibitors. So some mutations lead to a more sensitivity to the therapy, such as the common deletions of exon 19 or the point mutation at position 858. But mutations in the exon 20, such as T790M mutation, lead to resistance by changing the ATP binding domain and thereby inhibiting the interaction of the tyrosine kinase inhibitor. Under the tyrosine kinase inhibitor therapy pressure, the T790M mutation is often expanding, and for its early detection, a close follow-up of the patients is necessary. So here we designed SOP of the workflow using cell stabilizing blood collection tubes, providing then a day time interval until plasma is prepared, and the samples are stored then until further use at minus 80 degrees, and the procedure is ending up with next generation sequencing analysis. And here are some results of analysis shown. Please note that we have sometimes less than 500 nuclear copies available for our study. So the amount of DNA was quite low. And in some cases, in addition to the primary mutation, the resistance mutation were shown by analysis of the circulating DNA. We had, unfortunately, only from a few samples also tissues, and you see here that the frequency which was detected are higher than the frequency in the plasma samples, but the most important thing that we could really nicely identify the mutations in the plasma samples. So in conclusion, target multiplex PCR-based ultra-deep sequencing is not only a sensitive technology for detection of clinical relevant mutants in tissue samples, but also for identification of variants in cell-free DNA. And future approaches will focus also on gene deletion and amplification using the copy number variation tool, and maybe also future approaches will focus on gene rearrangements. We think that the mitochondrial genome is a sensitive target for ultra-deep sequencing in order to characterize heterogeneous tumors, and maybe also gives us some other possibilities to use it in diagnostics. And QIAGEN has now also designed their own mitochondrial genome primer set, so that it's also now commercially available. So at least I would like to acknowledge the people who helped me to do all this work, and that is in particular Ulrike Keutsch and Hannah Eichert, but also many other people like Wafa, who did a lot of the mitochondrial work. Isabel worked on the prostate cancer, Claudia, Karina, and Lisa worked on the copy number variation of the BRCA genes. And then I had also really a lot of help in the QIAGEN team. So they helped me a lot of in the bioinformatics, but also with other support. So, and I would like to thank you for your attention, and I'm happy to take your questions.
QIAGEN.
Sample to insight.